All right, good morning. My name is Bryce Stollemer, and I want to tell a story about Axon Erase, where we're pioneering a first-in-class approach to control pain at the source, right where it begins. So postoperative pain management is a $17 billion market driven by over 50 million surgeries annually in the US. Yet, despite widespread opioid use, we're still falling short. Where 80% of post-op patients receive opioids, but 75% still report moderate to severe pain. This means that even with opioids, pain is not adequately controlled for most patients. Worse yet, poor post-op pain management has long-term consequences. Up to 60% of patients develop chronic pain after surgery, turning a temporary recovery into a lifetime of suffering and opioid dependence. So this highlights a urgent need for a better solution, one that delivers long-lasting source-targeted relief without the risk of opioids. So today's pain management options fall into two main categories, but neither truly solve the problem. On one side, we have opioids, which blocks pain signals, but carries a high risk of dependence. Then there's Advil and Tylenol, which reduce inflammation, but offer only short-term relief. In the middle, we see options like Journavex, a pill that blocks a sensory ion channel, Experl, a non-specific ion channel like local anesthetic, and gabapentin, which dampens nerve pain by modulating calcium signals. These options only offer moderate and temporary relief, but don't really address the source. So this reveals a clear gap where current treatments fail to provide long-lasting source-targeted relief. And that's where our drug steps in, designed to deliver durable, targeted pain relief while minimizing opioid dependence. So our team has done this before in addressing nerve problems at the source. So the team are experts in SARM1, which is the primary executioner of axons, and previously was blocked this target to preserve axons for neurodegenerative disease indications, which led to a successful startup in Disarm Therapeutics and a significant exit to Lilly. And so that success just validates our approach and expertise. And now we're applying that same knowledge, but at this time we're activating axon degeneration by activating SARM1 to provide long-lasting pain relief. So long-term post-op pain is like a circuit that won't shut off. After surgery, when nerves stay plugged in, PEG signals continue firing, turning what should be a temporary post-op pain into chronic persistent pain. Other strategies try to stop this, but they fall short. Local anesthetics can temporarily block pain signals, but their effects wear off quickly, allowing the circuit to reactivate and pain to return. Meanwhile, opioids mask pain but don't address the source, leaving patients at risk of dependence and long-term consequences. So our breakthrough takes a different approach. We cut the plug before the pain starts by using selective axon degeneration to disconnect pain at its source, preventing it from becoming a long-term burden. So we're developing molecules to precisely activate this process through SARM1, delivering lasting, source-targeted pain control. And so our solution is designed with anesthesiologists and surgeons in mind. It works just like other long-acting intraoperational injections, but it is easy to integrate into standard workflows. But here's the difference. It selectively targets axons while preserving surrounding tissue, reducing toxicity and ensuring safer and more effective pain control. Our drug selectively targets and generates distal axons, which are the pain-carrying wires, while preserving the proximal nerve and the nerve cell itself. And so this disconnects pain signal at the source, turning off pain without damaging the nerve's ability to regenerate. And then relief lasts until the nerve regrows at 2 to 3 millimeters per day. As debris clears, regenerative cells rebuild the axon, aligning pain relief with the natural healing timeline. So to show how precisely our therapy works, we looked at its effect in an Evo mouse nerve model. So the results are striking. Only the distal part of the nerve, the far end, away from the cell body, undergoes degeneration, while the proximal segment, close to their body, remains fully intact. And so this selectivity happens because the drug only becomes active inside the neurons, limiting its actions to where it truly is needed. In the microscope images, you can clearly see the effect with healthy preserved tissue in the proximal region, and the target degeneration in the distal section. And also in a model of neuroma-related pain, this localized degeneration led to a drop in pain behavior, highlighting the potential of this therapy to offer lasting, focused pain relief without harming healthy nerve tissue. So we're addressing the growing demand for long-term surgical pain relief with a solution designed to last weeks to months. So our pricing strategies align with the reimbursement model for Exparel, a widely used long-acting anesthetic, and set it at 60% of its price to ensure favorable reimbursement and rapid adoption. So our initial focus is the long-term post-op pain market, starting with total knee arthroplasty as our beachhead. So This indication is ideal due to its high procedure volume, extended recovery, and uniform patient population, making it well suited for clinical trials. So success in TKA will position us to hopefully expand into broader orthopedic and cardiothoracic surgeries where long-term pain relief is essential. And so current pain treatments fall short. Exparel offers only short-term relief and doesn't meaningfully reduce opioid use. iovera lacks precision and raises safety and workflow concerns due to cold ablation. Zyme relief provides limited relief for a couple of days and also carries cardiotoxicity risks. And Alloy's ATX 101 lasts up to 4 weeks and is currently in clinical trials, but may be short for some certain surgical indications. But long-term bupivacaine safety is also remains unclear. So our therapy stands apart. It delivers targeted pain relief lasting weeks to months, preserves nerve integrity, can reduce opioid reliance, also while restoring healthy nerve function. So for our drug discovery, we will optimize lead compounds through in vitro screening, validate efficacy in an osteoarthritis model, and benchmark against standard of care. And then safety and durability will be confirmed in rodent and pig models with pharmacokinetic and pharmacodynamic data and also non-GLP tox data to support our mechanism of action and also safety. And so for our ask, we would need 5 million in seed funding to complete preclinical studies to finalize our IND package. We position us for clinical entry. This will de-risk the program and hopefully set up the stage for 45 million raised for Series A to advance through phase 1B trials with hopefully some strong early data to pursue a $70 million Series B round to power a phase 2 trial and prepare for strategic exit. So our team brings deep cross-functional expertise in neuroscience and drug development with a distinct edge as our leaders are among the pioneers in targeting the SARM1 pathway. We're not only advanced drug programs, but have successfully built and exited a company around this exact mechanism. So we bring kind of the full stack, scientific innovation, translational expertise, and operational strength to hopefully build and scale an impactful biotech venture. All right, thank you.